So, good morning, students and friends, and my dear colleagues, professors. And this is a fine morning, and so I would like to welcome our my dear student Prabu. So, I was a little overwhelmed when I meet after 24 years. We were communicating Prabu, Genomics director, and all. I was asking what is he and all about to Saranya. Later morning only I came to know that is my ward student, and I felt very familiar. Cool, right? So nice meeting you, Prabhu. And about Prabhu, so he's a really challenging guy. When I was interacting with him, I was able to experience that he did his UG, BSc Agriculture, during 96 2000 batch, and later he went to Madras University of Madras, Department of Biotechnology, did his MSc program there. And late afterwards, he went to Shivaji University in Maharashtra, and there he did his doctoral program. And during that program, I was able to see, I was going through his curriculum vitae, and I was able to visualize that yes, lot of publications during that period of time, a lot of impact factor, more than 30 publications in a high impact factor journal. So he's a research oriented guy, and he also did RNA research with Prabhu, sorry, this our Arul and all. He was working here during the MSc program. When you were doing after? Okay, okay. So RNA research and all these things. So, and he was under the staff exchange program of UK India Education and Research Initiative. He was he is a chief technical officer of the Indian Society of Certification of Organic Products, and he is the director of Syngenome OPC Private Limited, Coimbatore, and also the director of research and development of Srishti Biosciences Private Limited, Hyderabad. He has a facility in Hyderabad and also has a facility in Coimbatore. So he will be traveling between Coimbatore and Hyderabad very frequently. And when I was interacting with him, he is involved in product development and also exports, and he is giving and many services, Nanopore and services. He also brought that the thing. Instrument here, you can have a look and the students can have a look, and I think after this program you can also interact with him. So what I felt is we always teach in your campus the theory of it, but what he has achieved out of that knowledge that he has gained, he is now in a position to give more than for 50 people are working under him. That is the growth I can see. He is giving livelihood to more than 50 technology oriented guys, both agriculture division, biotechnology and all. So this is a nice opportunity for the students to also have an interaction with him to what you call is practically utilize for your future development, for your career, for livelihood or whatever it is. So that is the way. So it is a great initiative by the university. I should thank the Vice Chancellor for this initiative, because its professor of practice was not there previously, but now only it is there in all the departments where the students can have a industry oriented linkages. So that is a really a thing. So nationwide, this type of thrust is being taken up in all the universities. The industrial linkage is necessary because at the end, after your career, you should be in a position to take a venture. That confidence you should have. For that, this type of motivational lectures from the industrial people will be of very great help. So I take this opportunity to welcome our Prabu to this thing, and I think you will be gaining much out of it. Thank you for the opportunity. Thank you.
Okay. Good morning once again. Um, Before start of the talk, I would like to share my happiness that I'm with my coordinator and my teachers. During my UG studies, we had a course, Introduction to Biotechnology. It's a theory course. So this is a 2+0 credit. Dr. Madam and Arul sir taught that course. So because of them, I got interest in biotechnology and continue my MSc and PhD studies in biotechnology. Thank you, thank you, ma'am. Thank you, sir, for the short introduction about and other things. In 2019, I have started the Genome OPC Private Limited. In Genome, we are working on genomics, mainly related to medical field. And we develop NGS-based platforms for Nanopore and Inimula companies, like gene panels we used to develop for diagnosis purpose. And now we are involved in synthetic biology platforms for novel biological expressions. This is our core field in Genome. With that experience, today I am here to talk about Nanopore-based next-generation sequencing and its application. Before going into this Nanopore topic, I would like to tell a short intro about nucleic acid sequencing because most of them are students. So we all know that Sanger method of sequencing in 1977, that is the breakthrough in DNA sequencing field. So from that point, there are drastic changes and improvements that happened. And over the past years, from short oligonucleotide sequencing to millions of bases sequencing, from single gene to whole genome sequencing, and then from short read to long read method, from first generation to third generation. Now fourth generation sequencing also methodologies also came. In case of first generation sequencing, we used to get reads of less than 1 kb in length. But in next generation, like Roche and Illumina platforms, we are able to get massive parallel sequencing, and we can increase the DNA quantity per run. So, but there is a drawbacks in these two cases. As a solution, the third generation sequencing methodologies came, like PacBio, Nanopore sequencing. Among these two, Nanopore got wide interest in the sequencing field because it provides long reads and we can do real-time analysis. We can do single molecule sequencing also. This technology is developed by Oxford Nanopore Technology LLP from UK. It provides on-demand biological information by any person and in any environment with any sample we can do sequencing. So coming to the principle, how the Nanopore NGS works. It's a simple principle. If you take a cell or nucleus, there is a outer membrane. In that outer membrane, there is a protein nanopores, which forms a tiny holes that in nature form a gateway to the cell. By that, there is based on gradient, there is a molecule in and out of the cell. Movement is there. So that is the basic principle in Nanopore also. In Nanopore, there is a synthetic membrane is present. In that, there is a protein nanopore molecules are embedded. And this entire complex is floated in electrophysiological solution. And then there is ionic current is passed through nanopore. When the DNA or RNA molecules passing through this nanopore, there is a change in ionic current signal. So this change of signal is estimated, determined, and converted into nucleotide bases. So this is the working principle of nanopore technology in simple terms. So because of this, there are a lot of advantages in Nanopore NGS. So during sequencing, we do entire fragment of DNA or RNA molecule sequencing. So the read length is directly proportional to length of the DNA to be sequenced. And this read length is influenced by many factors like DNA extraction methodology, library preparation methods. For example, if you are doing standard lab extraction method of DNA, we used to get reads from 10 to 100 kb length of reads. And based on specific sample type, if we refine the protocols, and refined method of DNA extraction, we may get more than 2 MB size of DNA fragment sequenced. So by this, we are able to get long reads, which provides unambiguous reads. So that is easier for us to map with the reference genome, and is easier for us to assemble the reads, sequence reads. And another advantage is here we can sequence directly the DNA or RNA molecule. So there is no need of copy strands or synthetic strands, surrogate markers, and then there is no need of PCR amplification. Because there is no need of PCR amplification, we will not get error in the reads. So we will get amplification bias is removed. So coming to the steps in Nanopore NGS, from wet lab to analysis. In wet lab, in laboratory practice, we initially planned the objective of the study, sample type, and other parameters. From the sample, we will extract the DNA, and the DNA is subjected to library preparation for sequencing. And then we use these library for Nanopore instrument for sequencing. And after sequencing, we used to get the reads, raw reads. Reads means sequence informations. And in this, the analysis part is much more important. So in that case, the bioinformatics field is involved. So being the I think most of them are bioinformatics students. In this talk, I would like to concentrate on basic terms used in Nanopore sequencing data analysis. So the terms are base calling, data types, file formats, and what are the platforms are available for analysis. Base calling. It is a computational process of converting raw electrical signal into nucleotide sequence. Here the input type is POD5. This is the file type. POD means portable Oxford device file. And then the output we used to get is FASTQ file. And there are a few features, applications are available in this base calling. We can convert the raw signal into nucleotide. And if the sample is multiplexed by barcode sequences, we can do demultiplexing. And we can do alignment with the reference genome also. So these are the features available in base calling term. And coming to the data types, there are 3 standard data formats are available: FASTQ, POD5, and BAM. In this, FASTQ is the standard format that is the commonly used format in bioinformatics. It is the text format, file format. It is a small size. It occupies small size in computational storage. It contains base called sequence and their read score, quality score. It's a default format in bioinformatics. And POD5 and BAM is optional file formats. POD5 is raw signal, electrical signal. So if we want to do base calling, if any error happened, any mistake, any doubt is there, we can use this file because this is a large, it occupies large space in the computation. So this is optional file only. And BAM is alignment, genome alignment file format. It is also text-based. In this case, we can get the DNA modified base informations also. So based on the computational storage facility available with us, any one of these file type, data type we can store for the sample. So this is the FASTQ format of sequence. It is a text-based. It occupies small space. It has 4 lines for each read, sequence read. The header line, sequence line. And again, the header is denoted as plus symbol. And then 4th line is quality score of the particular read. So header line contains read ID, run ID, and sample ID, and then the channel number, nanopore channel number, which pore the strand is got sequenced, that pore number, everything will get in the first line. And the second line is sequence information. And the third is a plus symbol that denotes the quality. And then fourth line is quality score of each base, that is in ASC2 character. And coming to the file formats, so in bioinformatics analysis, all these file formats are important, starting from raw data to result file. And all the file formats are not in single way. Both the direction we can use it. Raw data is stored in POD5 and FASTQ. And alignment file is in BAM. And reference genome aligned data is in FASTA format. Annotation files are in GTF. And finally, we used to get result file in VCF. So with these terms, we'll come to analysis platform options. Nanopore provides two analysis platform. The one is Minno, the another one is AP2ME. These two are fully supported by Nanopore. And these two are compatible with any operating system, Windows, Linux, Apple, and any operating system is compatible. And to use these two tools, there is no need of bioinformatics expertise. These two are user-friendly. And other tools are developed by research communities. They used to develop the command line based softwares. So those are less supported by Oxford. And these tools are, to use these tools, we need bioinformatics expertise, because we should know commands, command programs suitable to Linux platform, Linux-based operating systems. So today I will cover these two fully supported analysis platforms. Minno, this is an on-device software. So there is no bioinformatics expertise is needed. Here we can do base calling, QC check, and analysis also. So in base calling, we can do real time. While sequencing itself, we can do the base calling. Base calling means raw signal to converting into FASTQ format, sequence format. And the post-run also we can do analysis. And there are models, accuracy models are available in base calling, super accuracy, high accuracy, and fast accuracy. In super and high accuracy, there is accuracy rate of conversion is high, and it takes more time and more computational space is required. Whereas in fast model, the accuracy is low and takes less time and less space is required. And we generally in industry, we use high accuracy model instead of going super. In that case, we need server kind of storage and all those things. In this base calling, Minno base calling, we can analyze and get the information of DNA modifications also, like DNA methylation, acetylation, those information also we can get it. This is the overview of Minno. So here the sequence are base called, and then is subjected to QC quality check, quality control check, based on the quality score, which we fix, and then is subjected to analysis. Here the analysis, the sample is barcode, we can do demultiplexing, we can do alignment with the reference genome, and we can do enrichment or depletion in case of adaptive sampling method of sequencing. And all these analyses are converted into simple shareable report format. So we have a final report. This is the system overview, working overview of Minno. Coming to AP2ME, this is a user-friendly end-to-end workflow. We can do in cloud server. There are three solutions. Cloud-based, that is cloud server, we can load our data. In that case, all the softwares are pre-configured and loaded in that server. So here we'll get the interactive and shareable simplified report. And then next solution is local. Means we can install this software in our laptop or sequencing device. Here we can, here in this case, we can do in the real time. We are able to do analysis after post run. So this software, in this case, the software is configurable. We can modify the software, and the output is shareable reports. And GitHub solution is command line based. We have to write the commands for run the analysis. So here the entire thing is user defined. We have to define the programs, options, parameters, everything. In general, cloud-based and local solutions are mainly used instead of command line. And this is the overview of Epi2ME working aspects. Here, the DNA sequence data is stored in local system files, and then it is getting uploaded in the software. And after uploading in Epi2ME, it's getting converted into base calling. And then data are real-time analyzed. And finally, we'll get the report and the reports are shared. In Epi2ME, there are many workflows, not only base calling, like quality check, barcode demultiplexing. We can do 16S metagenomics analysis, metagenomic analysis, microbial identification, and then classification also. Antimicrobial resistant detection also we can do analysis. There are separate workflows. Once we load the data, it gives the output. Automatically give the output. Once we set the antimicrobial gene database, all those things, if we set in the software, it will kill automatically, it will give the output. And we can do reference genome alignment also. So with this analysis platform, this is a basic platform. Remaining things are command line based. We need there's that analysis thing is wide and wide scope is there. So I'm not covering those command line because it's a preliminary, very preliminary to the students. And coming to application, Nanopore, we can use it by any person in any environment for any sample. It's a very hand-held instrument only, not like Illumina and other instruments. It's a very hand-held instrument. We brought that instrument also. There are 4 or 5 types are there. One is pen drive type of instrument. That is MK1B, we used to tell. And then another one is that a system was inbuilt with that analysis computer also. Entire computer is small packet sized. So that is MK1C. And then there are flow cells are different types are there. Depends on data availability for us. If you need short information like less than 2 GB or 1 million reads, we can go smaller flow cells. So for that, we have flunkel type of instrument. That flunkel get inserted into the MK1B. We can run the sequencing. And then bigger devices are available. Multiple flow cells we can run. Those things are involved in medical diagnosis. So coming to application is wide application, starting from gene sequencing to metagenomics, human genomics, cancer biology, and we can do genome assembly. We can do infectious disease diagnosis, target sequencing, and many more. So these are the few references. And the recent one, 2023 and 2020 is the short review of Nanopore technology. If you can study, go through that paper, you will get the entire principle, applications, everything is available in that review article. So thank you. We'll keep the instrument here. The students can view those things also. We didn't brought the flow cell because the flow cell is the main part of instrument, that is the costly one. The Nanopore devices are very economical. Around $800, we can get it a complete set with the 12 flow cells. Thank you. So it's open to discussion because many of them are students. Basic questions are there. Expand. Okay. Now, after my PhD studies, I worked in academic side and research aspect projects for almost 12 years. So like taking the class, running the research project, everything. And then 2019, I started the Genome Private Limited, OPC Private Limited. Genome is synthetic genome. That concept we started that company. It's a CRO kind of company, like a custom research organization kind. We used to get the custom research from other companies. We do the research, and the research output we will convert it into a product, and the product will give it back to the company. So they will brand it in their name. So in this case, we are not involved in market. Advantage for us, because many product development, many PCR-based techniques, everything is, there is opportunity for biotechnologists. So we did the synthetic biology platform for expression of all COVID-related protein antigens. And then we developed real-time PCR kits, and then LFA, lateral flow analysis kits for point of care diagnosis of COVID. This all started for research purpose, and then the output we converted into product, and then we sold it to European companies. So they marketed in Europe on their brand name. But we didn't get the license for selling in India. So that's a drawback. And then in continuation of that, we are expanding the NGS platform. Now we are developing gene marker panels for diagnosis, like prenatal diseases, arthritis, and sudden cardiac death. For that, we are developing a gene markers for a Nanopore platform. We are working for Nanopore. So we used to develop the set of panels. So they will make it as kit format. They will use that kit for sequencing with the patient samples. This is the part of Syngenome. We have a manufacturing facility in Hyderabad also. There we do medical diagnosis and diagnosis related reagent supply for the diagnosis labs like ThyroCare, micro labs. They have an instrument, they need the kits and reagents. Those things we'll do. And this is the main activities in our company. So here we are not stopping in academic research itself. We are continuing converting into product. It's a very simple. Most of the lab facility instruments are made by us. Like start with the gel documentation system, and then now we are developing microfluidic based devices for DNA extraction and this nanopore sequencing also. If we add the sample and kit reagent, it will mix based on the program and do the sequencing. So these simple ideas we are making into product. So this is the activity in our company. Now, the many factors influencing in this: sample type, and then extraction methodology, and quality of the DNA. If the quality of DNA is good, we can do within a day, because the DNA quality matters in output. If any impurities, any noises are there, and the output will not get expected result. Very low quantity of result will get. So the advantage in Nanopore is compared to Illumina. Illumina in case of Illumina, we'll use to get the short reads up to 300 bases. It makes the DNA into fragment and sequence, and we have to do assemble. Short read assemble will give many errors. So Illumina is also a good platform. The thing is depends on the study type. But whereas in the Nanopore, we used to get longer reads. So that is advantage in genome sequencing, genome assembly, and all those cases. If in case the sample genome is not covered in Nanopore, people used to do Illumina also, short read, and they will fill the gap when there is a mismatch. So in that case, they will fill the gap. So in that case, scientists will do both Nanopore and Illumina. And PacBio also available. PacBio is a bigger length will get, but it's economically cost. So Illumina and Nanopore is a better option. And analysis, coming to analysis part, in analysis, in our view, industry side, command line tools are the best one instead of user-friendly softwares. The thing is, in user-friendly software, if you load the sequence, we don't know what will happen. We'll get the output. So if any error in the file format, it will overcome and run continuously. It will omit that particular part and overcome. So we'll miss those important data. Whereas in command line, it's difficult to learn. If by practice we can learn easily, but in command line, the process will stop in the step itself. If any file format, any line is error, anything has happened, it will stop there itself. Will rectify and go. So in that is the advantage in command line tools. So that is the reason in bioinformatics, most of the advanced tools are command line based. No, Illumina has got developed earlier than Nanopore. That is the only platform available early days. So people preferred that one compared to Sanger and other methodology. Now the Nanopore workflows are developed. So now it got reduced compared to Illumina. Reduced. Illumina it is. Many kits they have developed. And then many platforms are available. Workflows are available. Workflows means steps, analysis steps and library preparation steps. And they are doing the research on that topic also, and they are coming out. So we can't say Illumina is bad. Illumina is one of the technique. Nanopore is one of the technique. Lesser coverage. Yes, yes. Yes, coming days it will come. It will come near to the Illumina pricing and lesser than price also. Illumina price also. In Illumina case, we are getting the short reads. So in Nanopore, we are getting long reads, and the coverage definitely will be lesser. But in analysis part, it is easier. Long read assembly is easier. And if once the DNA quality is better, we'll get the good result and good depth. In both the case, Illumina and Nanopore also. It's a general criteria. This is followed in lab. Like for before starting the DNA extraction, the sample should be preserved. It should not get degraded or deformed. All the storage condition we have to follow. So the initial extracted DNA matters. For that, all the steps precautionary steps we have to take. So that is the main consideration. The remaining thing, library preparation, everything is like PCR kind of. You'll take the DNA, mix the reagent, and load into the machine. That's all. But the DNA quality is important. For that, we have to follow all precautionary steps. In that case, that is more precaution we need because RNA will get degraded. Now many kits came, for example, to avoid the degradation and RNA is out. Many things are available. We have to use those things. And in general practice, normal lab practice, we have to store properly. And -20, -80, we have to store the RNA molecules. In Nanopore, we can, no need of converting the cDNA. We can directly do the RNA sequencing. So that case, the PCR bias will not come. amplification error will happen in PCR. That will not come. And we'll get the 5' end and 3' poly A tail, everything will get in Nanopore. But in cDNA library preparation, we have to use the primers, poly A tail primer, and we will not get cap, 5' cap, mRNA cap. It's much more technical terms are is there. So if sir permits also, we are ready to give the training also, hands-on training also with our instrument and other thing. You can use your own sample. Thank you. Thank you. Thank you. Thank you, sir. Thank you.
Good morning to all. So on behalf of the university and also on behalf of CPMB, we would like to express our deep sense of gratitude to our Vice Chancellor Madam for giving us permission to conduct this kind of professor of practice. This is for the first time and the university we are conducting this, inviting the industry people so that the students will get benefited, so that they will in turn, they will become an opportunity to become an entrepreneur. So with that, I would like to express the coordinator, Madam, Dr. Saranya, for inviting Dr. Prabu from Genome. And also we would like to have a special thanks to Dr. Prabu for coming over here to deliver this interesting lecture to our students in this fine morning. And also, I'd like to thank the Professor Biochemistry and other faculty members of bioinformatics and biotechnology for attending this lecture. And also, I would like to thank the students for hearing this lecture patiently. And once again, I thanks to all. And also, I would like to thank our director and dean SPGS for motivating us to conduct this kind of lectures. Thank you, one and all. Thanks for the opportunity.